Okay. So just as a recap, we were going off on the types of movement that lipids can do through a cell membrane, and talked about rotation, talked about lateral movement that they can just kind of wander around through. They can also flip-flop between the internal and external side. The thing that I was trying to reinforce there is that this flip-flopping doesn't happen necessarily evenly. Certain lipids will not flip-flop as much as other lipids will. Also, moving for any particular lipid, flipping from outside to inside or inside to outside are not equal probability as well. So it's not that any given lipid is going to flip one way more than the other way. And we'll talk more why that's important in just a few minutes. But despite this, so you have all these things moving around. You have proteins that are embedded in here moving around as well. So again, just kind of reminding you that we have this, hey, I have a thing and I can do a thing with a thing. That's pretty cool. So coming back here, remember we have these proteins that are kind of lodged in here. And to some degree, they're going to be floating around as well. So everything, we don't have like a strictly solid membrane. It is somewhat liquid. However, that being said, it's not that you get movement such that everything just evens out throughout the entire membrane. Things don't just kind of float around until essentially diffuse through the membrane until everything's equally distributed. In fact, we actually have heterogeneous patches in the membrane, things being held in certain locations for good reasons. And let me give you one example of that. And that would be lipid rafts. So a lipid raft is an area of the membrane that tends to be more ordered than the surrounding membrane. So if you look here, this area 2 right here, this is a lipid raft. And here we just kind of have the regular kind of normal membrane. So lipid rafts are interesting because, as I just said, they're more ordered. And these lipid rafts, come back to that, tend also to be more tightly packed than the surrounding membrane as well. What happens is that you get more saturated fatty acid tails in the lipid raft than you do outside the lipid raft. You tend to have more unsaturated fatty acids outside of the lipid rafts, which means that the raft itself becomes more tightly packed than the membrane around it. In addition, there's more cholesterol that's in the lipid raft itself. That's represented by these little, I don't know, they look like little one-legged men to me. So there's little one-legged men that sit in the lipid raft. It sounds like a really sad raft. I don't know, they could be women too, one-legged women, I don't know. But there's these little one-legged people that are known as cholesterol that are sitting in the lipid raft. And that's the glue that holds them together. The cholesterol is interacting with the surrounding phospholipids in order to help essentially stick that part together. But also, there is a lot of sphingolipids in these lipid rafts as well. The sphingolipids specifically are interacting with the cholesterol and also, through that means, also helping to hold this lipid raft together. So we have this relatively tightly packed cholesterol and sphingolipid-rich lipid raft that hangs together, and they're not as fluid as the surrounding area because it's more tightly packed. It tends to be more crystalline, less fluid, and so these components that are in the lipid raft will tend to hang together and not just diffuse out into the rest of the membrane. So we commonly use these in signaling pathways. Oftentimes in a signaling pathway, and we're going to talk a lot more about this as we get on down in the quarter, is that you have many membrane proteins that need to be close to each other in order to interact with each other. And so you'll have, like, for instance, a receptor, it may be a G protein, a few other associated proteins, and they need to interact with each other so that you can actually carry out whatever signal, transduce whatever signal you have from the outside to the inside, and amplify that signal. Well, if all of those components were just kind of willy-nilly floating throughout the entire membrane, the chances of them interacting would be really low. It'd actually take a long time for these signals to either be transduced through the membrane or you would have to have such high concentration of those proteins that you would run out of space for anything else. So, in order to prevent both of those problems, you can pack them all together in a lipid raft so they're relatively close to each other. They can interact with each other easily, and you don't have to have so many just all throughout your membrane that is kind of a waste of energy, waste of space. So, throughout the membrane is not homogeneous. The makeup is not homogeneous kind of as you go throughout the membrane. However, this is also true as you look on the inside and the outside. The bilayer is not symmetrical inside to outside. It is somewhat asymmetrical. And there is a tendency, now I just want to really point out that I'm going to give you a tendency of the two, the intracellular and the extracellular layer, but these are not completely strict rules. These are tendencies that tend to be this way. There tends to be more phosphatidylcholine, sphingolipids, and glycolipids on the extracellular layer of the bilayer. Tends to be. It's not exactly that all the time. Glycolipids is one thing we're going to come to. There's a lot of those on the extracellular layer, largely because these are important in cell signaling. The intracellular layer tends to have more phosphatidylethanolamine and more phosphatidylserine on the inside. Now again, this is partially a consequence or maintained because that flip-flopping inside and outside is not equal probability for all lipids. If it was, these differences inside to outside would very quickly go away because essentially all the flipping inside and outside would even out the concentrations inside and out. That's not what happens. Okay, so that's the membrane components. Now that we're done with that, are there any questions on membrane components, anything that we talked about up until this point? They have parts, and they do things. That's what we're going to talk about next, them doing things. So, we also have proteins in the membrane, and these are what carry out most of the functions that the membrane is going to carry out. Now, we're going to get a lot deeper into the functions in a little bit, but right now we're just going to talk generally about the proteins. We have, as I've talked about already, we have extrinsic or peripheral proteins. These are essentially equivalent terms. They're not exactly equivalent terms, but for the purposes of this class, they are. Close enough. And again, extrinsic would be those, they're just sitting not completely through the lipid bilayer, but are just kind of sitting on the outside here, or on one side. It can either be the intracellular or the extracellular side, it doesn't matter, but they tend to be just on one side. And these, we first figured out what was transmembrane versus what was extrinsic or peripheral proteins by figuring out how hard was it to remove it from the membrane. The peripheral or extrinsic proteins are easy to remove. We can actually change the pH of the solution that they're in, or just change the ionic strength. This will somewhat deform the protein a little bit, and we can pop it out of the membrane relatively easily. So this is how we first started categorizing them, is that you could change the solution, pop out all of the extrinsic proteins, pellet them out, separate them from the rest of the membrane, and then start characterizing them. We now know that's true. We could do that because of how they're situated in the membrane. We also have the intrinsic or integral proteins. These are the ones that go all the way through, like this. Those are stuck in there a lot better. These can be single, double, or multi-pass. What I'm talking about in this case is that generally, remember that our proteins are just a string of amino acids. So we just essentially have this string of amino acids that's going to fold itself up into some sort of 3D conformation. When we're talking about a membrane protein, this chain is going to pass several times, at least once, through the membrane. So imagine you have your membrane here. I'm just going to, that's my membrane. Got different colors up here? No! I want all the colors of the world, but I only have this color. Okay, that's fine. And so you might have a membrane protein that just goes once through the protein. Or you might have a membrane protein that just goes once through the membrane, like that. That would be a single pass. You only have the amino acid chain passing once through that. So this would be a single pass. Alternatively, you could have one that comes through, it turns around, and comes through again. That, which would be double pass. Or, one that comes through many more times, which would be multi-pass. Which every time I say that, it reminds me of The Sixth Sense. Anyhow, or I mean, in this case, it would be a 4-pass. You have 6-pass, 9-pass, 10-pass. Oftentimes, these are going to be arranged. Sometimes they can be arranged in things like barrels, where you actually get you have alpha helices that alpha helix goes down, alpha helix comes back up, alpha helix goes down. And if you're looking at it from the top, you have like alpha helix, it comes over, and then it's alpha helix, comes over, it's alpha helix, comes over alpha helix, alpha helix, alpha helix, like that. And it creates a channel through the middle of that. A lot of transmembrane channels are 6-pass alpha helices that have this pore in the middle that allow things through. These are a lot more difficult to remove. You have to add detergent to actually pop them out. The reason for this is that you actually have to disrupt the membrane. Detergent is just something that is also going to be amphipathic, and detergents do a really good job of disrupting cell membranes. And so you disrupt the cell membrane, you have to do that to actually pop these guys out of the membrane. Continuing on with carbohydrates. So all eukaryotic cells have some carbohydrates on the cell surface. All of them do. And that whole collection, the entire collection of all the carbohydrates that are associated with the surface of a cell membrane is known as the glycocalyx. A calyx can be referring to something that is covering something else, glyco referring to the sugar. So it's like the sugar coating. You didn't know that all your cells were sugar coated. You were sweeter than you thought. Although don't eat yourself, that wouldn't be good. You're not in that way. So the glycocalyx has quite a few functions. One of the most important of those, or the one that is the most common, is for cell recognition. The major way that cells recognize other cells and what they are is by interacting with carbohydrates on the cell surface and then figuring out by some signaling pathway what that cell is. You find a lot more about that if you take immunology, because in the grand scheme of things, a lot of your active immune system is cells recognizing other cells. They also have a lot to do with membrane stabilization. So the glycocalyx can do a lot to help stabilize and reinforce the membrane. And the interaction, really this comes through an interaction of a lot of the different carbohydrates together. So you have covering carbohydrates, they're interacting with each other and helping stabilize that membrane. So these can get attached. These carbohydrates aren't just existing by themselves on the cell membrane. They're attached to things. And so we have them on lipids, making up glycolipids. And glycolipids can have anywhere between, generally have one carbohydrate chain. You don't always have one carbohydrate chain. They take a carbohydrate chain, stick it on a lipid, and these are usually stuck onto neutral lipids, but not always. You can also have them on sphingolipids or many others, but neutral lipids tend to be the most common. And we also have glycoproteins. So again, carbohydrate chains that are stuck onto proteins. Almost all proteins that are in the cell membrane have at least one carbohydrate chain. Nearly all. Not all, but nearly all have one. Many of them have several to many carbohydrate chains on them. So, nearly all have at least one. Many of them have several. So just to kind of come back to this illustration, we have a glycolipid here. This is just a neutral lipid with a carbohydrate chain on it. We have carbohydrates sticking on glycoproteins here, or on proteins, making them glycoproteins, and on and on. So, one example of this is the ABO blood grouping that you have on your erythrocytes, or your red blood cells. This largely depends on the specific carbohydrate type that you have on a specific protein on your red blood cells. It specifically is on what's called a band 2 protein, and you have carbohydrate chain on that band 2 protein. It's called band 2 because one of the very first thing, I mentioned that one of the very first ways that we were figuring out what's where on the cells is we're like trying to pop all these proteins off and figure out where they go. Doing that by changing pH and detergent. Well, one of the very first things we were doing that with in humans was blood cells. Blood cells are really easy to collect from people, and unlike, your brain cells or something else like that, you can usually take a lot of blood, some blood, and it doesn't really mess with the person so much. And so we were doing this and they're just easy to work with. It's easy to work with just a straight solution of only blood cells. You can usually separate it out from the other parts of the blood fairly easily. So we're popping those off, and then essentially electrophoresing all of the membrane proteins out, and a lot of the membrane proteins on your blood cells became named for just like where they fell out in these electrophoresis gels. Like, oh, hey, this is a second band. It is band 2. Has nothing to do with what it actually does. So on this band 2, it's a glycoprotein. If on that specific glycoprotein, the terminal carbohydrate is an N-acetylgalactosamine, you are type A blood group. If on that protein, on the carbohydrate chain, the terminal carbohydrate is a galactose, then you're type B blood group. If that does not have a carbohydrate chain on that particular protein, you are type O. And then you're type AB if you have both. You have some proteins that have the N-acetylgalactosamine, some of them that have the galactose. Any questions about that? Yeah.
Is that attaching to a glycoprotein?
This is a glycoprotein. Specifically, these are connecting to a glycoprotein. The protein is called band 2 specifically. Band 2 happens to be an ion channel or a transporter. I think it might be a bicarbonate transporter. I don't remember exactly off the top of my head. Okay, let's begin talking about the functions of the membrane itself. What kind of things is the membrane doing? And one of the major functions that happens at the membrane, other than just keeping things separated, of course, is keeping things separated, but it's also involved in transport. You do want to carry very specific things across the membrane. You want to be able to pull in certain things. You want to expel certain things. And so transport is one of the major functions of the cell membrane. It's also really involved in signal transduction. We want to take signals from the outside of the cell and somehow get the signal inside the cell. Generally, we are talking about this happening with no actual individual molecule passing across the cell membrane, although that can happen as well. But just the signal itself, the information in some way getting from the outside of the cell to the inside of the cell. And then also cell recognition, being able to identify another cell. So let's begin with the first of these and talk about cell transport specifically and see how does that happen. And there's a couple different ways that it can. The first off is just simple diffusion. Things can just simply diffuse right across the cell membrane. No need for transporters, no need for channels, just diffuse right across. Now, you might think, hey, that seems to defeat the purpose of having a membrane if things can just diffuse right across, and maybe to some point, but there's actually very few molecules that can do this. There's not many. And actually the ones that can do this, it's actually nice that it can do this. So they have to, if a molecule is going to float across the cell membrane, just diffuse across it, it has to have several characteristics. First off, it has to be hydrophobic. The interior of the cell membrane is hydrophobic, so to get across there, it also needs to be hydrophobic. Also, it has to be small. And this is kind of reiterating what we said before, but also has to be uncharged. So a small, uncharged, hydrophobic molecule. If you're big and hydrophobic, it does not matter. You're not getting through. If you're small and charged or small and hydrophilic, you're also not going to be able to get through. And also, it has to be down its chemical gradient. In other words, there has to be a lower concentration on the other side of the cell membrane than the one it's coming from for there to be net movement of that particular molecule across. You can actually might still move across, but there won't be net movement one way or the other. And for this, we're not relying on proteins to get across. You don't have to have specific transporters for this. So one molecule that's really common to think about that does this readily is the gases that we exchange across the cell membrane. Oxygen and carbon dioxide. They're both small. They're not charged. Relatively hydrophobic-ish. And they just readily pass across the cell membrane. And this is kind of nice because then you don't actually have to have, like, for instance, oxygen transporters in your cell membranes or carbon dioxide transporters. It can just diffuse straight across your cell membrane, get out, get in. It's a little bit more complicated in the grand scheme of things with carbon dioxide, but it's definitely true with oxygen. Yes?
Oh, I have a quick question.
Yeah.
Uncharged.
Uh-huh.
Like, why can't they be charged? What would they interact with?
So, well, first off, you're going to interact with the polar head groups of the lipids that you're going across. And so your polar head groups have charges on them. And if you have charges on you as well, you are going to be interacting with those charges, and that's not going to work real well. Any other questions? Yeah, Justin.
Back to the blood groups. Is there any benefit to having a different blood group, or is it like other than donation, like O can donate to people and stuff like that?
Well, if you're AB, you can get blood from anyone, so that's kind of nice. I mean, no, not. I don't know of any specific other than exchanging. I don't know of any specific biological benefit. And that's probably why all of them exist in the world pretty commonly, especially like A and B. Those are pretty common. so it doesn't seem like there's very strong reason, biological reason for one indicator in this. Yeah. O tends to be relatively uncommon, but I don't know if there's any good biological reason for that to be. I think AV is uncommon as well, but again, I don't know if there's any biological reason for that. No. All that's a long way to say, I don't know, I don't think so. Yes?
Can you explain a little bit more about what you mean in down chemical gradient?
Okay, so essentially what we're talking about is your chemical gradient is how the concentrations are between where you're coming from and where you're going. So let me just give you an example. Let's look at the case of oxygen just for. Let's say we have a cell membrane here, and we have. And I'm going to just talk, since I'm talking about gas, I'm going to just go talking pressures here. So let's imagine I have inside the cell, I have a pressure of, let's say, 12 kilopascals. Yeah. Of oxygen here is your pressure of oxygen here. So, and then out here, I have something like 16 kilopascals of O2. In short terms, assuming that like temperature and things are the same across here, there's more oxygen here than there is here, which means that you're going to tend to have them moving this way. This is your concentration gradient. There's higher. I mean, technically, I'm telling it to you in a pressure gradient. Let's do it this way. Let's say there is 400. No, let's keep it 16 kilopascals of oxygen here, 12 kilopascals here. Just because I know this one is okay. Kilopascals. Okay. But yeah, that means there's more oxygen here. So it has to go from an area of higher concentration to lower concentration. It's not going to, in net, go from lower concentration to higher concentration. In truth, you might have one stray molecule going across here and there, but the net movement is always going to be from higher concentration to lower concentration. That's down this chemical gradient from a higher of the gradient being from high to low. We have a difference in the concentration. And so it's going to move from areas of high concentration to low concentration. Does that make sense? So if we look at diffusion, it does this. If we look at the rate of diffusion on the y-axis here and the concentration of the molecule, or the gradient of the molecule, as we get a higher and higher concentration gradient, the rate of diffusion is just going to go up and up and up. That will go on essentially forever. I mean, in truth, there's probably some limit to where just surface area is going to start. You're losing surface area, but for the most part, that just continues to go up forever. That won't be true for all of them. There is also another kind of transport that we call channel-mediated diffusion. In this case, we're still relying on diffusion. It's just diffusion moving something across. However, we have some sort of protein channel through the membrane that allows those molecules to get through. And so, for instance, we'd have something like this, where we have a channel, essentially a protein that's a several-pass, a multi-pass protein, and it has some sort of pore in it that allows some molecule to move through, some set of molecules. Or something like this. If you're looking at it from above, oftentimes these are 6-pass alpha helices, not all the time. There's a lot of different configurations, but it seems pretty common. You have 6-pass alpha helices, where you have some pore, then in the middle that's something like, say, sodium. A happy sodium can move through. I want to be the Bob Ross of biology. There's a happy sodium. Talk to the sodium. Make friends with the sodium. Something like that. So, like I said, these are generally multi-pass alpha helices. And primarily, we're going to get ions through there. Like I was talking about, sodium is a common one. Sodium, water, potassium, other small inorganic ions tends to be what we get through there. Bicarbonate, other random things. We have a small opening in there, usually 1 nanometer or less in diameter. It's very small. And again, these ions, because this is diffusion, they still have to move down their electrochemical gradient. Now, I'm talking about electrochemical gradient here because, in this case, we're charged now. And so we have to worry about 2 things at once, whether or not we're going to move spontaneously across the membrane. We both have to worry about the concentration of other things that your same molecule inside. So if I'm a sodium, I am not going to move across the membrane if there's more sodium on the inside than the outside. But you also have to worry about the charges on each side. If I'm a positively charged ion, and it's more positively charged on the inside than it is where I'm at right now, that's going to inhibit me wanting to move across the membrane. So you have 2 things to keep in mind here, both the electrical gradient and the chemical gradient, when figuring out whether or not a charged ion is going to diffuse across the membrane. This wasn't the case in simple diffusion because we're just talking about uncharged molecules in that case. Oftentimes here we're talking about charged molecules. Oftentimes, as an aside, these proteins are going to have some sort of selectivity filter on them as well. You have some sort of apparatus on the protein here that makes it so only a very specific type of ion will move through. So only sodium is going to move through, only potassium is going to move through. And it does not allow anything else to go past. Yeah?
So what would be an example of positive charge inside the cell keeping positive sodium out?
I will actually tell you, I have a very specific example of this coming up probably today, if not early tomorrow. But just giving you an example, let's imagine, okay, out here we have a sodium ion, and he's happy out there, but there's lots of sodium ions out here. And inside there's very few sodium ions, but there is potassium ions. This sodium ion is going to tend to be pushed, for lack of a better term, inside because of its concentration gradient. The concentration gradient tends to want to push it in here. But all of these positive charges, and we're just thinking about the chemical gradient, it doesn't matter that these potassiums are in here because they're not sodiums. It doesn't matter for the chemical gradient. But because they're positive charges, they will tend to oppose the sodium wanting to come in here because it's the same charge. And so if you have enough positive charges, if this inside is more positively charged than out here, then that will oppose that from coming in. And what you end up having is you have to figure out which force is stronger. Yeah?
Does one of those factors tend to trump the other, or does it just depend on like the different concentration?
It depends on the different concentrations. So, for instance, almost all cells have some sort of electrical potential in that their membrane, essentially all cells in your body, have a difference in charge across the membrane. This gets really exacerbated in things like neurons. Neurons have very, very strong differences in electrical potential across the membrane. And so in neurons, the electrical potential actually is a lot stronger than the concentration gradients. In a lot of cells, however, and that is also the case in muscle cells. So in muscle cells, you also have a very strong potential, and the electrical potential is really important. In a lot of other cells, like in a liver cell, although it does have a potential across there, it does maintain a difference, you're probably, for most cells, going to have a concentration gradient that's going to be more important. Although, that being said, it depends on how strong the concentration gradient is. If you have a fairly weak concentration gradient, the electrical gradient could be greater. So really, it depends. It's both of these are important. The times that you most often see it as being a really important thing, like more important than the concentration gradient is in electrically active cells like muscles and neurons. Any other questions on this so far? So, like with simple diffusion, with carrier-mediated diffusion, it follows the same pattern. As we increase the concentration gradient, or we increase the concentrations on one side of the molecule, the rate of diffusion is going to go up, 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 essentially indefinitely. Now, an interesting thing with this is in channel-mediated diffusion, these channels can be gated. They can have two conformations, an open conformation and a closed conformation. What this often looks like is something like this, where you have a channel. This is in cross section here. You have your membrane here, and a lot of them, what you have is, for instance, like in sodium channels, essentially an inactivation gate that you have like this little thing that it can swing like a little ball on a chain that can swing into the pore and essentially physically close the pore. Some others of them essentially have a different conformation where you have a narrowing in here, and in certain conformations of the channel, this narrowing will actually close off so nothing can get through. So there's a couple different ways in which gating can happen, but essentially it's just you have a channel, you have a protein channel, in which in different conformations it's either closed or open. It can either allow the passage of molecules through or not. And this can actually be gated for a number of different reasons. It can be ion gated, in which the presence of a certain ion binding to it can change that conformation. Yeah. Oh. It could also be voltage gated. It could be looking at the voltage or the relative potential between the two sides and change the gating as well. We can also have ligand gated channels. A ligand is just some generic term for any molecule that's going to bind to a protein. In this case, we just have some molecule that might bind to a place on this channel, let's call it a square, and will either open or close this channel. This is essentially how most neurotransmitters work. You essentially have an ion channel, some ion diffusion channel, and then your neurotransmitter is some ligand that's going to bind to that, open it up, and allow the passage of some charged molecules to go through that channel. Yes?
What's the point of having a voltage gate if there's already the electro diffusion?
The reason for that, okay, so your question is why do you care about having a voltage gate if you already have a voltage difference that could be opposing that? Yeah. Essentially, what you could do is it's like having a dam that opens up. Like, you don't want just the voltage gradient to control it. So what often happens? So one comment is coming back to neurons, because voltage has a big deal with neurons. What they can do is they will build up a huge, both chemical and electrical gradient, both going in the same direction into the cell. So essentially what happens is the inside of a neuron tends to be net negatively charged. The outside of a neuron tends to be in the extracellular space net positively charged, and you have this huge concentration of sodium on the outside. And so what happens is there is this huge both chemical and electrical gradient wanting to push sodium to the inside of that cell. So you have channels, sodium channels, voltage-gated sodium channels that are on the surface of that neuron. And what's going to happen is you're essentially holding back this huge gradient until something happens that changes the voltage here and then allows the sodium to come through. Now, the thing that changes the voltage is actually could be a gate down here opening. And by allowing all of the sodium down here to come through, it's changing the electrical potential across that membrane. Does that make sense? But then that kicks off this one because it's voltage gain. Now this one allows all the sodium to come in, changes the voltage, and it propagates down the cell membrane in that way. Does that make sense? So essentially you're holding back. You don't want it to go down as electrochemical gradient. You want to hold it back until you get the signal and that signal is voltage. It just happens to be usually a voltage of one next to you. Good questions. Next, we have carrier-mediated diffusion. So this might sound very similar to channel-mediated diffusion, and it is somewhat similar. But the difference with carrier-mediated diffusion is that instead of just going through a channel, you actually have a molecule binding to a transport protein. It has to bind to it, and that protein is going to deliver it to the other side. And like all of the diffusion that we have talked about so far, this needs to be, again, down its electrochemical gradient. What that means is that going down its electrochemical gradient is going to have a negative delta G. Remember just for a second here, when we're talking about delta G's. This might confuse you just for a second, but remember, you might think to yourself, how in the world can you have a negative delta G? Because essentially what we have here is we're thinking about this like an enzyme. We have a substrate plus an enzyme. Going to like the enzyme substrate complex, and going to the enzyme plus the substrate again. It's just that the substrate is now inside the cell, whereas here it was outside the cell. Now, it's not exactly an enzyme, but we can think about it essentially the same way. If you're thinking about this, you might think to yourself, well, hey, this and this are exactly the same. So how in the world do we get negative delta G if that's the case? So let's think about this for a second. Remember, we have the delta G naught, which is the standard free energy. And so the standard free energy for this and for this, you were right, are going to be exactly the same. But the standard free energy takes into account a lot of different things, including the temperature that both of these are at, but one of the other big things is the concentration of the reactants and the products. Let me back up. The standard delta G doesn't take those things into account. The standard delta G is standard, but to figure out the actual delta G, the actual free energy, you have to take into account the concentration of the reactants and the products. This is at standard conditions. This is like at 25 degrees C, at very specific molarity, all that. You then have to take into account the temperature and the concentration of the reactants and the products and everything else to figure out what the actual delta G is. So if you remember, for instance, when we were calculating up, was it last week, how much energy is released by the hydrolysis of ATP, I gave you a standard free energy, but then I gave you a different free energy. It was like 11.3 kilocalories per mole, whereas the standard free energy is like 8 something. That's different because the physiological conditions in which it occurs are different than the standard conditions in which we figure this out in the lab. Does that make sense? So under those different conditions, it's different. So you're right, this and this have the exact same standard free energy, but the environment that they're in is not the same. What happens here is the environment will tend to be high concentration of reactants. And this is going to tend to be low concentration, or not tend to be, it'll have to be, low concentration of reactants. Or whatever S is. Which means that the G here is going to be higher than the G here. So this transport can have a delta G if the place that the substrate ends up is a lower concentration of that substrate than where it started out at. Does that make sense? Now this is important because this isn't just something flowing around. This is actually now, in this case, you have a protein that is going to, you'll bring that up here. We have a protein that is going to have a binding site for a specific molecule. is going to essentially pick that up, carry it to the other side of the membrane, and toss it out there. And so you might say, hey, that's doing work. It's doing something, but we're not paying for it with ATP. And that's right. We're not paying for it with ATP. You pay for it with the negative delta G that happens from taking this thing from a place of high concentration to a place of low concentration. But this has a consequence. Because we have a molecule that has a binding site, that you need to bind the molecule you want to transport across the membrane to it. As you increase the concentration of the molecule that is diffusing, you'll go up and up and up like in the other ones for a while, but you will top out. You will reach some sort of asymptote. It will not increase the velocity of diffusion infinitely. The reason for this is the binding sites of those carrier molecules will get used up. It's like the difference between using a hose and having to carry something somewhere. I'm not exactly sure what you would send through a hose that you would also carry. I know that analogy was poor. I'm going to carry this water in my hands. I guess you could. Onto my lawn and go. I'm going to go get more water in my hands. Watering my lawn. I spent all day watering my lawn. What's that? Oh. I didn't want to do that. Okay. Really quickly here. There's two major models of how this might happen. There's the old one, which was the rotating carrier model, which was prevalent into the late 70s. And essentially what was here is that you have this carrier transport molecule, this carrier protein in the cell membrane. Your substrate will bind to it, and it rotates, and it drops it off on the inside. So then rotate and dump it off on the inside. That's how you get it working. The new model is we have what's called the alternate conformation model. And this is essentially what we're using today with a few modifications. And essentially what we have here is an idea that we have a binding site that alternates between a high and a low affinity state. And also the entire enzyme or the entire carrier alternates between inward and outward facing state. So, for example, in this, here we would have the enzyme as a whole is in the outward facing state, and the binding site is in the high affinity state. So it's likely to bind said substrate. And so this comes in and binds readily. The binding of the substrate then changes the conformation of the protein to be the inward facing state, and the binding site then changes to the low affinity state. And so it drops it off on the inside and dumps it out. Okay, we will pick up all the cool ways we can get things across the membrane on Friday. Until then, enjoy the rest of your Wednesday. Get sleep because sleep is good.